When we try to understand a wild ecosystem, we rely almost entirely on what we see. We scan the tree line, index the canopy, and point our lenses at the vast landscape. Our eyes are naturally drawn to the giants. Take the shoebill. It's a 5-foot-tall predator with an enormous, unmistakable beak and piercing yellow eyes. Or we spot a Madagascar pond heron in the moment it's swallowing a massive frog. These are the dramatic events of survival that end up in documentaries and field guides. Because human beings are highly visual creatures, this is how we've cataloged nature for centuries. We identify a species by its color, by its shape, and its distinct physical markers. But if we drop our gaze from the canopy and look into the low scrub of the African savanna, that visual toolkit stops working. Down here, you find a tiny, streaked brown bird. In a different patch of grass miles away, you might see a bird that appears physically identical, perfectly matched to the golden brown stems. These are members of the Cisticolidae family, and there aren't just 2 of them. There are roughly 160 distinct species, scattered mostly across all the warmer regions of the Old World. Despite this variety, nearly all of them share the same drab brown or gray appearance. This creates a challenge for traditional biology. Our primary method for exploring the wild relies on spotting physical differences, and here, those differences are often invisible to the human eye. The reason for this physical uniformity comes down to habitat. These birds nest low to the ground in wide-open grasslands and scrub. If you are an insect-eating bird the size of a mouse living out in the open, bright red feathers or an elaborate crest will immediately attract a predator. In this environment, extreme camouflage is a survival requirement. But if 160 species are all wearing the same brown camouflage, they still need a way to tell each other apart to find mates or defend territory. They solved this by shifting their species identification system away from sight. Instead, they rely on sound. To our eyes, a winding cisticola and a trilling cisticola are difficult to distinguish, but to their ears, the pitch, rhythm, and cadence of their calls act as distinct biological signatures. In the wild, an observer identifies a cisticolid the same way another bird does, not by sight, but by recognizing the specific acoustic signature of its call. This acoustic system works for the birds, but for decades, the complete lack of visual markers confused the scientific community. Because early taxonomy was based on physical traits, researchers grouped these birds into a massive catch-all category called the Old World Warbler family, or Sylviidae. This classification remained standard until 2013, when researchers prioritized molecular data to verify the family's true evolutionary history. Researchers used multi-locus phylogeny. By analyzing distinct genetic markers across the bird's DNA, they mapped the actual relationships within this visually identical group. The results revealed a family tree far larger than realized, branching into 26 distinct genera, including prinias, apellices, and tailorbirds. These findings establish that these visually uniform birds represent an ancient, diverse evolutionary radiation. If you go on an expedition to the wetlands of Uganda, a successful biological survey looks a bit different than you might expect. Biologists and expert guides regularly log over 500 bird species on a single trip. They don't achieve that number just by chasing giants with a telephoto lens. They spend hours standing completely still in the dense brush, mapping the ecosystem entirely by ear. True biodiversity is not always colorful, and it doesn't always show up ready for a photo op. There are massive branches of the evolutionary tree that are hiding right in plain sight, camouflaged as simple little brown birds. To fully grasp the complexity of the natural world, we have to recognize the limits of our own eyes and learn to tune in to the frequencies the wild actually uses to communicate.